Hello, welcome to Biology with Omotayo. The next subtopic under our main topic, Introduction to Biology, is scientific method and the use of the microscope. Scientific method basically deals with the systematic approaches of making investigations with regards to inquiries that arises from observed natural phenomenon. So, scientific method. As I said much earlier, that it has to do with the systematic approach of making investigations with regards to inquiries that arises from observed natural phenomenon. That is to say, scientific methods only commences from the act of observation, and so in rolling out the order of events concerned with the use of scientific method, the first is observation. Observation simply means the use of human senses to reach a particular deduction. It could be the eye, it could be the tongue, it could be the skin, and so on and so forth. Basically, senses are used to obtain details. Immediately after observation, the next is identification. For identification, it simply means the act of determining the correct placement of a particular deduction. The act of determining the correct placement of a particular deduction. So it does involve classifying, grouping, making inferences, and so on. After identification comes hypothesis. Now, hypothesis, simply put, is an intelligent guess. So, from what you observed, you were able to identify and classify based on your experiences. Now, you make an intelligent guess, a postulation. Okay, intelligent guess, hypothesis. And it doesn't end there. After hypothesis now comes what? Experiment. Experiment is to test hypothesis. Remember, hypothesis is an intelligent guess that can be tested with experiments. An intelligent guess that can be tested with experiment. And now comes experiment. Experiment helps to verify hypothesis. So that simply means that after experiment, there must be something else. And that is what? Results. The results from experiments will determine if hypothesis will be accepted or rejected. Which means after results, there is something else. And that is what? Theory. Theory is a extensively tested hypothesis that is proven to be true. Extensively tested hypothesis that is proven to be true. That is theory. So this theory can equally be further subjected to more study. And so this brings us to laws or principle. So a well extensively further tested and proven to be true theory results in a law or a principle. These are guides to all that is required for necessary deduction to be made in science using the scientific method. Now, I will connect all this using a chart. Remember, we started from observation, then identification, next hypothesis. Now, the hypothesis will guide us to design our experiments. After designing your experiment, you conduct your experiments. After conducting the experiment, surely you will obtain a result. Now, these results can either be accepted or can be rejected. Here, if the result is accepted, accepts results. That simply means you will progressively move to theory and then to law. However, if our result is questionable, you come back here and you reject results. You reject the results. Then you have to redesign experiment. You have to redesign your experiment, and then you conduct the experiment again and now see if your result will now be acceptable. Sometimes it may equally require that you equally look as well into your hypothesis. So at other times, it may be required that you don't just redesign experiments. It may require that you revisit your hypothesis and then you redesign. Okay? You redesign your experiment and then you conduct the redesigned experiment and watch out to see if your result will be acceptable. Afterward, you will now be able to progress to theory and then finally to law. Thank you. Quickly, the second aspect. We talked earlier about looking at scientific method and the use of the microscope. So, the microscope is of concern because it is an instrument that is used in making further investigation, especially when dealing with organisms that cannot be well appreciated or seen with the naked eye. And basically, it is used to ensure magnification, enlargement of this organism. It is an instrument that helps us to view more clearly organisms that are smaller than 0.001 millimeters. Organisms smaller than 0.001 millimeters. And basically, the microscope is made up of a couple of components. To start with is always the eyepiece. So, to start with is always the eyepiece, the lens for viewing the magnified object. I can put a rough sketch here. So, we are starting with our eyepiece. That is where we are starting from. The next is the body tube, and this is my body tube. Okay, the body tube provides attachment for the eyepiece. My eyepiece attached to the body tube. Afterward, we have the revolving nose piece. So I come this way, and I have something like this. This is my revolving nose piece. So eyepiece, body tube, revolving nose piece. Now, on this revolving nose piece, we have our objective lens attached to it. So you have objective lenses. They can be rotated for you to carefully select the preferred lens that will enable clarity with regards to the organism being viewed. So our objective lens is the next here. Objective lens. The objective lens. Now, after the objective lens, we equally do have the arm. Okay, this is my arm. Okay, the arm do help in handling the microscope properly. Remember, we are dealing with lenses and you need to be careful. Now, towards the upper part of the arm, we do have two knobs. A bigger knob and a smaller one. The big one is usually referred to as the quartz adjustment knob. The quartz adjustment knob, while the small one is always referred to as the fine adjustment knob. Okay, now the quartz adjustment knob is for focusing the organism to be viewed at under low power, while the fine adjustment knob is for focusing organism to be viewed under high power. From here, we'll be making progress. We can see another part of the microscope being depicted. Okay, this is the stage. Okay, the stage where we will mount our specimen or stain slide, as the case may be, for viewing. And on your stage, you do have clips to hold your slide in position. Here, you have a regulator for opening and closing your condenser. Yes, the condenser helps to regulate the volume of light that gets to the specimen being viewed. So it has a regulating arm here that helps to either open it further or reduce it to regulate the volume of light that gets to our specimen that is mounted on the stage. And as you can see here, for a mirror microscope, what you'll be having here will be a mirror. For a light microscope, there will be a light bulb that will be producing light ray or beam, as the case may be. And this beam is being, the volume of it that gets to our specimen is being regulated by the condenser. So with this, I guess you have a better grasp of the microscope as an instrument used for investigating inquiries with regards to the study of biology. If you have any question with regards to scientific method and the use of the microscope, kindly drop your questions at the comment section. They will be attended to. See you next class. Do have a great day. Thank you. From the intricate working of the cells to classification of living things,